Hi, this is Akib Hassan Ali Khan from University of Burgos, and currently I'm working as a postdoc at ICRAM in the field of environmental biotechnology. Today, we will going to discuss about transcriptomics and how we can use transcriptomics to study cell-cell and cell surface interaction, and what are the possible implication of this particular field in this particular area. Before moving to the specifics of this presentation, it would be good to revise the basic concepts in the in the molecular biology. That is, a particular function is being coded by our genes, which is present in DNA, and to code a particular protein, they have to make messenger RNAs, which will ultimately translated into the proteins. And if we wanted to see how the environment is impacting the organism and how a particular condition is impacting the organism, there are numerous ways through which we can study. One of this way is to study the transcriptome, or I say the messenger RNA transcribed by the microorganism or any other organism in a specific condition, because it gives us the indication that how a particular organism is responding to the molecular to the environmental stimuli or to the environmental condition. If we talk about transcriptomics, it basically gives us the information about the expression of all the RNA, whether it is coding or whether it is non-coding, at a specific condition of time, because of certain active changes that happens because of the environmental stimuli, or because of the stress, because of any other interaction that could happen to the organism. So we can say that studying transcriptomics will give us the snapshot or snapshot of the expression of the microorganism that is happening. And with this transcriptomics, we can analyze not just messenger RNA, but all the non-coding RNA that are present within the microorganism, or maybe a higher organism. There are numerous ways through which we can use transcriptomics, but in general, if we say that if we wanted to study RNA, that would be qualitative or that could be quantitative. In qualitative way, we are just analyzing the expression of certain gene, and then in quantitative, we are analyzing the level of expression. So we can study if a particular gene is being expressed, or in general, if a particular gene is expressed to a certain level. Both of these answers can be given by can be answered. Both of these questions can be answered by using transcriptomics. There are different ways through which we can perform transcriptomic studies. We can perform differential gene expression among the microbial community. We can even go for the single cell transcriptomics to study all the metabolic changes, or you can say transcriptomic changes within a particular cell. We can even analyze metatranscriptomics, in which we can analyze multiple microorganism or multiple organism within the population or within the community of microorganism. Or we can even go for ribosomal profiling, where we can see the active microorganism present in a particular time. So with this different type of transcriptomics, we will be able to answer the changes in the biological responses. We can even study the cooperation and competition within the microbial community. We can even analyze at what specific time, what different type of antibiotics or what different type of specific biological molecules are being expressed or are to be expressed, and we can analyze different tolerance, let's say, against a particular antibiotic or any other how we say environmental stress. So if we talk about the microbial colony or microbial association, there are numerous stages through which it exists in its life. If we see how formation of a biofilm, they usually exist in the form of planktonic bacteria that get attached to a particular surfaces, and they started to self-aggregate or auto-aggregate, ultimately producing extra polymeric substances that lead in the association with other microorganism, which develop a particular biofilm, and then ultimately it get mature with the passage of time. So these mature biofilms then act as the sanctuaries for the microorganism to further colonize in other places. Similar type of growth is also present in eukaryotic organism, for example, with fungi and algae. When they make association and make lichens, and they perform lichenization as they find the favorable condition, ultimately making thallus and then maturing as a thallus, and then this thallus is acting as the sanctuaries for the growth of other organisms. So seeing at each specific point, whether it is the attachment, whether it is the maturation, whether it is the dispersal, microorganisms usually undergo numerous changes, and then ultimately the transcriptome analysis at each specific time will be different, and they will be expressing different type of mRNA or messenger RNAs or the proteins ultimately. So analyzing these transcripts, analyzing these transcriptome will give us insight for numerous things. In general, we can say that these biofilms will help us understand the biodeterioration or the bioprotection of certain surfaces. Biodeterioration can occur with the form of clogging of the pipes, dismantling of the surfaces, or maybe puncturing of the surfaces because of the growth of the biofilm. And bioprotection can happen by the prevention from the environmental stresses, for example, excessive rain, excessive, how I say, sun exposure, and all these can contribute toward the bioprotection. But in general, if we understand how these microbial life exists at each specific stage, we will be able to understand the development, the functioning, and the overall development of this microbial biofilm. So with transcriptomic analysis, we will have the idea how the microbial attachment works, what are the mechanisms involved in it. We will have the idea how microorganisms are interacting with each other. We will also have the idea that at specific point of time, what will be the expression of genes, what will be the specific phenotypes that are possible with the exposure to certain stimuli. We will be able to identify how we can prevent the development of these microbial biofilm, and we can also selectively promote the development of certain microbial films or biofilms for our own purposes. So having said that, it is very simple and it is also very difficult to perform transcriptomic analysis. So the main upstream challenges that can occur is this, that RNA is quite an unstable molecule compared to DNA and protein, and hence care should be taken in account when we are extracting mRNA or any type of RNA from the sample. There is always sample heterogeneity, so it should be carefully kept in mind that the RNA of interest that we are collecting is extracted from a particular mattress, and there could be a possibility that the size of RNA extraction is the size of the sample that we have is extremely small, and then because of the heterogeneity of the sample, it would be very difficult for us to extract the RNA from a particular given sample. Then there is a problem of quality. The extraction of RNA is totally dependent on the time at which we extract the RNA. It is highly dependent on the method that we adopt for the extraction of RNA. It is highly dependent for the environmental condition in which it was extracted. Technical variability can occur, and then there is always possibility of interaction with the other extracellular matrices. For example, there are different type of compounds present on the extracellular surfaces. As soon as the cell rupture and we are extracting the RNA, it can make complexes and then can result in the reduction in the availability of free RNA. So having said that, it requires care to be taken at the upstream level of the RNA extraction. Even if we see what are the ways through which RNA transcriptomic analysis has been performed in the passage of in the last few years, there has been different technologies which were being used for the analysis of transcriptome, but the major one include RNA-seq, messenger RNA microarray analysis, and express sequence tags. And among these, the use of RNA-seq has been gaining in interest with the passage of time. There are numerous reasons for it. Could be this, that it is not dependent on the availability of it. We can analyze RNA without the availability of genome. It could be any other. But then if we see the pattern of publication, if we see the pattern of utilization of research, people are more, or researchers are more opting towards RNA-seq compared to the type of RNA transcriptomic analysis. So if we talk about the pros and cons of each of these technologies, so they all exist. For example, for RNA-seq, we do not have the problem of accuracy and sensitivity because it is highly sensitive, it is highly accurate. We can also detect the novel transcript. For the drawback, if we talk about the messenger RNA, RNA sequencing for the transcriptomic analysis, it has high background noise, so we required upstream, downstream processing of the data that we receive from the RNA-seq. There is possibility of high quality RNA requirement, because if we do not have high quality RNA, we cannot perform RNA-seq, and ultimately upstream problems can check the benefits of RNA-seq. Similarly, if we talk about RNA microarray, the throughput of this technology is comparatively fast and very, how we say, instantaneous compared to other technologies. But then again, it is highly dependent on the availability of prior information of the transcript, and without it, it cannot perform the hybridization, and that is the basic of this microarray analysis. For the express sequence tag, it is also quite low cost, and the problem associated with it is also quite similar to the mRNA analysis. So for the purpose of this presentation, we will be discussing the messenger RNA, transcriptomic analysis using RNA-seq. For RNA-seq analysis, if we somehow manage to get the high quality of RNA, we require to do the RNA fragmentation using the process of sonication or biochemical enzyme treatment, which result in the RNA fragment, and then this RNA fragment is used for the production of complementary DNA, and the complementary DNA is then used for the production of second complementary strain, and then the second complementary strain can be another additional telling, which is a poly-A telling, depending on the type of sequencing method that we use, and ultimately the adapter are being ligated to these reads, and then these ligated reads are used for the amplification and sequencing. As soon as the sequencing is performed, we usually receive the information in the form of FASTA-QC file, and these FASTA-QC file can be used for the purpose of analysis. But before moving towards the analysis, we require to perform the quality check and data normalization of our data that we achieve using RNA-seq, and for that there are different softwares that we can use to check the quality of our the sequence data that we receive. There are different software like FastQC, and for the normalization technique, there are different normalization technique like RPKM or FPKM, which is a way through which we can perform the normalization of data. And now, depending on the type of RNA-seq data that we receive, whether it is having, whether the genome of this particular data is already available, or the genome is not available, or the data is coming from the microbial community, we require to go through different processes. For example, if the genome is available, we can simply go with the quantification and abundance. After performing this, we can simply go for the differential gene expression. But if the data that we are getting do not have a genome already available, then we need to go for the de novo transcript assembly. And for this, there are different software like Velvet, StringTie2, or Salmon. We can use this for the assembly of only one type of organism. But when we are dealing with the microbial community, then we need to perform an extra step, which is called binning. And in this, we just make the clusters of different communities all together, and then can see how the gene expression are happening within each of these communities. And then ultimately the differential gene expression or functional annotation can be performed. We can even perform what genes are co-expressed, what type of genes are down-regulated, what type of genes are up-regulated, what are the pathways that are responsible for a particular process, and we can even go for the functional gene annotation. So that is quite a brief introduction, or you can say information related to the use of transcriptomics and how we can use it for cell-cell and cell surface interaction. And for more detail, I would recommend you to go and explore the resources that were presented by SurvBio project, and more detail is also available there. Thank you.